Thank you all for joining us today. This is a special presentation with the Sustainability Series, and it's also funded partially by the Dreyfus Foundation. That's who gave you the treats outside and provided travel for Dr. Karen Anderson to be here. Karen Anderson obtained her PhD from Ohio State University. She is now at Yale University, and she holds multiple positions. She's professor of pharmacology and molecular biophysics and biochemistry. She's also the co-director of the therapeutics and chemotherapy program. She's involved in teaching undergraduates and graduate students about drug discovery and structure-based drug design. Her research focuses on understanding on a molecular level how enzymes play a critical role in such diseases as cancer and infectious diseases, including AIDS, which is going to be a focus of her talk today. This is the first lecture, which she is also giving another lecture tomorrow at 3 p.m. in the Sokol Room. And also there's two other events. There's a Women in Science breakfast at 9 a.m. tomorrow in the Center of Computing and Information Science Skyway Lounge at 9 a.m. And there's also a question and answer with students and lunch from 12:30 to 2:30 in Richardson 365. So join us for tomorrow for more events. And now, without further ado, Dr. Karen Anderson.
Thank you. So I'm honored to be here. Can you folks in the back hear me okay? Okay, good. I'm honored to be here today, and as you actually heard a little bit from the introduction, my lab is interested in sort of a multidisciplinary approach using structural, molecular, and synthetic techniques, and especially mechanistically using rapid reaction techniques. And our strategy is really to dissect enzyme mechanisms or even look at receptor-ligand interactions. And our goal here in getting this very fundamental information is to think about how to use that information for inhibitor design, and ultimately to think about how we can develop therapeutics. And so we have a number of ongoing projects in the lab, and so this just really summarizes several of the more projects that we've had ongoing for a number of years in cancer. And actually tomorrow I'll tell you a little bit about our anti-parasitic project. But today I want to tell you a little bit about our studies on HIV reverse transcriptase, and thinking about it especially from the standpoint of structure-based drug design. And that's actually the focus of today's talk, is going to talk a little bit more about the challenge of developing therapeutics. And what are we really thinking about in terms of developing AIDS therapy? So first of all, let's think about the epidemiology. These are some of the numbers for AIDS. We know still that it's still a worldwide epidemic. There are, as of now, over 37 million people infected with HIV, and there are, roughly 1 million to 2 million new cases a year. And the problem is that there are currently no cures. There's actually one person, as you guys have probably heard of, someone called the Berlin patient, who's been cured by a stem cell transplant. But there really aren't any cures yet or vaccines. And so the problem is that if an individual is controlled with HIV, they have to take therapy lifelong. So every day for the rest of their lives, to keep their viral level suppressed, they actually need to be on HIV therapy. And so we need to think about continually developing new therapeutics. And so what I've illustrated here in this slide is the HIV viral life cycle, and the step that we've been interested in is illustrated here, the reverse transcriptase step. But what I've listed here are some of the different steps in the overall life cycle that have actually been utilized by pharmaceutical industry to develop a number of drugs. And as you can see, reverse transcriptase really comprises a very large number of the drugs, and that's actually that step in which a single-stranded RNA copy is converted by reverse transcriptase into the double-stranded DNA, and as we know, that double-stranded DNA gets integrated into the host cell genome. Now, in order to manage the disease, we actually treat the patients with what's called combination therapy, and so in order to keep the viral load suppressed and protect functioning the immune system and the functioning of T cells, we actually need to administer more than one drug. And this is all combination therapy is usually administered orally, and it's, as I mentioned, something where the individual needs to take therapy every day for the rest of their lives. Now, to think a little bit about, from the standpoint to go to the molecular level, if we talk about reverse transcriptase as in a structural sense, this is a three-dimensional structure of reverse transcriptase. It's actually described as a structure like many of the polymerases as a right-hand structure, so you can think about it as having a thumb, a fingers, and a palm domain, and so the catalytic site actually resides in the palm. This is a three-dimensional structure solved by Steve Harrison's group a number of years ago with the catalytically poised conformation, and actually Tom Stites at Yale was the first person to solve a structure, and that was actually in the presence of an allosteric inhibitor, and I'll kind of get back to that in just a second. But the enzyme actually is a heterodimer, so there's a P66 catalytic subunit and a P51 subunit that plays primarily a structural role. Now, if we think about therapies and those that are actually approved by the FDA for treating, for targeting reverse transcriptase and for treating AIDS, they actually fall into two chemical classes, and you chemical folks in the audience will recognize that these look like regular nucleoside substrates, so endogenous substrates, and so the way they work is they compete with the endogenous nucleotides. They get phosphorylated to the triphosphate form, they get incorporated into the growing strand of DNA, and then because they don't have a 3' functionality, they actually result in a chain termination event. On the other hand, the non-nucleoside reverse transcriptase inhibitors, or NNRTIs, are actually chemically quite diverse in terms of their chemical scaffolds, and they actually target an allosteric site in the enzyme. And so the next slide illustrates where these actually reside. So that allosteric pocket, and that was actually an allosteric inhibitor that was in the first crystal structure that Tom Stites solved. That pocket is interestingly about 10 angstroms away from the active site. So the nucleoside drugs would bind and compete with the endogenous DNTP, whereas the allosteric site is really the site for the NNRTIs. And so that modulates catalysis about 10 angstroms away. Now, in order to, some of our very early studies really wanted to dissect the mechanism. I mentioned the fact that we really want to try to understand the enzyme first and then think about inhibiting the enzyme. And so I just want to mention the approach that we use, which can be very informative if one wants to get detailed mechanistic information. I know that you're all perhaps from your biochemistry classes familiar with steady-state kinetics, and in fact I had the pleasure of visiting one of the biochemistry classes this afternoon. We saw a lot of nice steady-state kinetics there. The approach that we use is called transient kinetics, and the important part of this is that this is certainly important to get a first look for the mechanism, but this gives you a handle for reactions occurring at the active site of the enzyme, and importantly, it allows you to monitor both the chemical rate of chemical catalysis as well as whether or not there might be conformational changes that are associated with catalysis. Now, as you might imagine, enzymes turn over very rapidly, and so if we want to get information that's occurring at the active site, we actually need to be able to look at the enzymatic reaction on a very rapid time scale. So we use a special piece of equipment to do these kind of experiments. And so some of the early mechanistic work that we did used a strategy called rapid chemical quench. And in order to look at the reaction on a very fast time scale, this is just an example where we would pre-incubate the DNTP with metal in one sample loop, and then we would load our enzyme and radio-labeled DNA substrate into the other sample loop. And then with this instrument, so we load the samples in, the instrument gives you a precisely controlled stepping motor that drives the two reactions together. So the reaction occurs here, and then the reaction is terminated upon reaching a quenching agent. Because these are metal-requiring enzymes, we use EDTA. So we simply collect the sample and we analyze it as illustrated here. And this is just an example where we've taken a radio-labeled primer template, a DNA, and this is a 20-mer, and we're incorporating DCTP. And so what you see here, if you run a long sequencing type gel, is you see the incorporation to form the 21-mer, and you see that this reaction is occurring much faster than 1 second. So you obviously need a special piece of equipment to do this. So one can do this kind of analysis, and you can get a number of key pieces of information in looking at inhibitors and trying to understand how nucleotides are incorporated. And one can, as I mentioned, get mechanistic information. So a pre-steady state burst would give one an idea of the rate of chemical catalysis, or the K polymerization, as well as a steady state rate. And then we can look at that rate as a function of DNTP concentration, or in this case DCTP, and you can define the incorporation efficiency, Kpol over Kb, and that really gives you a handle on how either substrates or the nucleoside type inhibitors interact with the enzyme. And so our very early work really delineated the mechanism for reverse transcriptase. And what we found in looking at the mechanism is that there's tight binding of DNTP, an initial tight binding of DNA, and then an initial weak binding of the DNTP, a rate-limiting conformational change as the correct nucleotide is bound, and then the very fast rate-limiting conformational change that governs the K of polymerization, rapid chemistry, and then the overall rate-limiting step in the reaction is the release of the elongated product from the enzyme, the elongated DNA product. And so when one does pre-steady state, we can actually get a handle on these particular processes. So we use that kind of strategy not only to understand the mechanism, but to understand how these inhibitors work. And so one of the interesting things that we wanted to try to understand very early on was how these allosteric inhibitors worked, and remember I showed you from an early slide that the inhibitor binds about 10 angstroms away from the active site. So how does it really control catalysis? And so what we found is that when these NNRTAs bind in this allosteric pocket, they actually change the overall rate-limiting step. So now that rate-limiting step is actually the chemical step. And so that gave us a handle to really understand how these enzymes work. But as I mentioned now, we know more about the enzymes, so we wanted to think more about therapies. And so we really need to try to stay ahead of developing new therapies, and some of the challenges that I've listed here, even though there are very good drugs on the market and HIV patients are living a normal lifespan, which is all very good, and it's significantly improved the quality of life for these patients. However, we know that reverse transcriptase is a highly error-prone polymerase, and so that allows the virus to very rapidly mutate and produce drug-resistant mutants. So it's like hitting a moving target. We have to try to stay constantly one step ahead of the drug resistance. So there are certainly toxicity issues that are associated with the current therapies, particularly if you're on the therapies for multiple decades. You start to run into problems. And so these are sort of some of the reasons that we need to develop new therapies. And so our lab is actually working on several different aspects of this in terms of thinking about molecular mechanism, drug resistance, thinking about drug design, and also understanding the molecular basis of toxicity. But what I'd like to tell you a little bit about is our work in trying to design new inhibitors. And so this is just some of the current landscape that we have for the NNRTI drugs here. And the current NNRTI drugs, such as efavirenz and rilpivirine, are really components of these single tablet regimens that patients take. So they take these daily. And rilpivirine has also been one of the agents that's been developed very recently into a long-acting formulation that was approved just a couple of years ago. And there's some suggestion, this is actually given by injection, that this could last for a month. And so that's one of the advantages if you only had to take the therapy as an injection once a month. And then there's actually a new NNRTI that was very recently approved by Merck. So these are some of the developing NNRTIs that are on the market. But why did I mention rilpivirine? I mentioned some of the very good things, and these are some of the good things with rilpivirine. It was one of the more recently approved, with the exception of the newest doravirine by Merck. And so it's certainly a very potent compound, and it's active on a whole panel of drug-resistant mutants. However, it actually has some problematic properties in terms of its pharmacokinetics and its ADME-tox properties, as I've illustrated here. It's not a very soluble drug, and so it's very difficult in terms of formulation. It has very poor solubility. It has a high log p-value, and we know in the drug discovery world that we actually want a nice balance between being somewhat water soluble and somewhat aqueous soluble. We like for drugs to be below 5. And so that's a problem. Also, it inhibits cytochrome P450 3A4, and there's the potentially reactive cyanovinyl group, and those are potential problems with toxicity, especially if you're on other drugs and you have the potential for drug-drug interactions by inhibiting this drug metabolizing CYP3A4. And then the most important issue with rilpivirine is the fact that there's a restricted dose limit so that you can only use it in patients that are diagnosed with a low viral load, under 100,000 copies. And that's oftentimes not the case for diagnosing a new patient, which will have more than a million copies, so their viral load will be very high. The other thing is that, and the reason that you actually have to restrict the dose is because it actually inhibits the hERG ion channel, which can actually result in cardiotoxicity. And so in order to approve it, they actually had to get the FDA to approve it at a lower dose. So that's sort of the ugly part of rilpivirine. And so then I want to tell you a little bit about our strategies to develop new drugs. And so this is our computational approach that we have been involved in with Bill Jorgensen's lab, who's in the chemistry department at Yale and has a computational group. And so the whole idea is, let's think about building in these very good properties in the chemical scaffold as we're developing the drugs, and then optimize those drugs. And so he's developed a series of computational programs that will allow us to look at our target site that we want to probe, and then we can either build potential compounds de novo, or we can actually search through known chemical databases of compounds or chemical scaffolds that are commercially available, and then we can filter those compounds by what we call an ADME-tox filter, okay, so that we throw out the bad things ahead of time. We want good solubility properties, and we want to throw out things that might be potentially reactive. And then we synthesize or purchase those compounds, assay them, and our goal just in an initial virtual screen is to get a micromolar hit. And then we can get some information, and then hopefully through free energy perturbation scans, and I'll tell you a little bit more about that in just a second, But that really guides us for synthesizing the compounds, again, filtering, and ultimately, hopefully, ending up with nanomolar leads. So that's sort of the strategy. Try to build that in. And so then our workflow for this, and this is where my lab gets involved, is we partner with the computational evaluations by using structural information. They do the virtual screen, and then we're the experimental part where we evaluate the screens both biochemically as well as cell culture, and we look at their activity on both wild type HIV as well as the drug-resistant mutant forms, and that's sort of the workflow. So the way one would go through one of these virtual screens is illustrated here. We would look at our target site. This is just one of the examples using a chemical library called the Maybridge Library, and doing a similarity search. This is a strategy where we wanted to try to get some novel chemical scaffolds, and we wanted to try to find some compounds that might bind both wild type as a drug-resistant mutant, kind of narrow those compounds down and select those, and then filter them for their admitox properties, and then ultimately ranking them and evaluating them. So we kind of go through this workflow to get some initial hits. And then in my lab, we actually have a number of strategies for assaying the compounds, and those really guide lead design. Those in vitro studies that I mentioned to get an idea of whether they inhibit. We can do detailed kinetic analysis, such as the experiments that I just showed you. We have some fluorescence assays where we can get binding information. Structural studies, and I'll mention a few of those in just a minute, to really help us refine our computational models, and then also to experimentally determine their physiological and pharmacological properties. And finally, the highest bar here is the ability of these compounds to have antiviral activity in human HIV-infected cells. So that's sort of the biggest bar. And so our assay is illustrated here. And this is actually an example where we set up the tray both in the absence of virus as well as in the presence of virus, and then we actually add the drug in increasing concentrations, and if our cells are still healthy, we see a nice purple color as illustrated by interaction with this MTT dye, as illustrated here. It turns purple. If we kill the cells, in the places where you see yellow are from the HIV infecting the cells and killing the cells. And so we get two important pieces of information here. If we do it without virus, this gives us an overall measure of cellular toxicity, which is important. We don't want something that's very toxic. And when we can rescue the cells so that the HIV is no longer killing the cells, we get an EC50 value, and we really want to look at that ratio between the EC50 and the cellular toxicity, or the therapeutic index. And we get really excited when we incubate our plates for several days, and we see purple not only in this row, but purple here, and then maybe we would even have to dilute that out. We get very excited, and that tells us that we have a potent compound. Now, in order to try to develop new scaffolds and put new functionality on the potential compounds and working with chemical scaffolds, I wanted to show you a view of the allosteric pocket, which is illustrated here. And what you see here in this space-filling model is there's plenty of room here for functionality to sort of build this in thinking about doing inhibitor design. Now, if we're using this whole strategy of something called free energy perturbation scans, what we would do, and this is my computational collaborator, would look at a potential scaffold. This is just sort of an example of this scaffold that we discovered from a virtual screen. And this only has a 30 micromolar, it's a 30 micromolar hit. And so in order to really guide synthesis, he actually computationally puts this in various positions and lets the computer decide which are some of the key compounds that we should make first, and then we make those, and it's sort of an iterative process. And so this is just an example where you can see a big effect in changing the functional group. So we can go very quickly, we just simply put a methyl group, substitute the hydrogen with the methyl group, we go down to 2.8 micromolar. If we substitute then a chlorine, go down to 200 nanomolar, so now we're in the 200 nanomolar range, and then if we include a methyl and a chlorine, we're down to 39 nanomolar, so we're getting a very potent inhibitor, and these are actually numbers that come from this HIV assay with infected cells. So this just shows an example. Another example here where we take heterocycle, we actually change even the positions of the nitrogen in the heterocycle, and we go from 15 micromolar to 200 nanomolar, and then further by putting a furan moiety on here, we go down to 6 nanomolar. So this is sort of the strategy, and we've used this over the last several years to guide and make a number of compounds, but what I'd like to focus the attention on is this very recent example where we've taken this diphenyl methane type scaffold with the terminal uracil, and we've developed these very potent picomolar compounds. So they're very potent, and in this particular case, we've been able to significantly improve that potency. And so, as I mentioned, the strategy here is really to do this in an iterative way. In this example, where we started with this docking virtual screening hit of 5 micromolar, and we were able to improve that over 90,000 volts. So that was sort of the starting point for some very, very potent compounds, and that's really what we've been working on to further evaluate and improve these compounds. And so this is just an example how we got to that point. I mentioned that we started out with a 5 micromolar hit, and this is just the impact of changing that functionality. If we start to put chlorines on, as I mentioned, with this chlorine scan to try to improve potency, we can go down to, sort of the mid-nanomolar range. Then we put a cyano group on here, we can further improve that. So this is the pathway toward getting very potent picomolar inhibitors, putting a cyanovinyl group in this position, and these are actually 2 of our very early key compounds that have picomolar activity in cell culture. So at this point, we had done a lot of computational analysis, and we had done a lot of biochemical and cellular analysis, but we really did not have the 3-dimensional structure. We only knew what was computationally predicted. So we really wanted to develop a strategy to determine the 3-dimensional structure of these compounds. So this is sort of our strategy and our protocol for getting the crystal structure of reverse transcriptase bound to these non-nucleoside inhibitors. We use this workflow, and so the whole idea is that we would validate the computational studies. And so for those of you that do crystallography, these are just some of the data statistics for each of the compounds. These are some of the very potent early compounds that we were able to get the 3-dimensional structure. And so what I'm going to illustrate on the next slide is how close we were to the computational prediction. So this is one of the early papers from the structures of a former postdoc, Kathleen Fry. And this just shows how these inhibitors bind. This is this catechol diether moiety, and we can see that it fits very nicely into this hydrophobic pocket. And this is just an example where the cyanovinyl group sort of points inward in the tunnel region, and it's flanked by tryptophan 229, proline 95, and other hydrophobic residues, and it interacts very nicely, the uracil interacts very nicely with the backbone lysine residues 102 and 103 here. Now, the teal is the actual experimentally determined structure, and the yellow is actually the computational prediction, and you see that we're actually pretty close in terms of the actual structure. There's a slight change in position, and there's actually a change in rotamer here with tyrosine 181. But in general, the computation very closely matches the prediction. And the value of the actual 3-dimensional structure is we can now use those for our for the next set of computations. And so we've been involved in further improving this class of compounds. And one of the things that we have done is we wanted to think about getting rid of this cyanovinyl group because that's an alpha-beta unsaturated type compound that could potentially serve as a Michael acceptor. So what we would not want to happen is a nucleophile reacting here and covalently modifying the protein. So we found that we could actually change this to this indolazine type structure, and that actually still binds very nicely into the active site, and we still maintain our picomolar inhibition of the enzyme. And so the next slide just summarizes some of these important properties, and what you want to see is green in all of the categories, and compare. What I've done here is compare these to the drugs that are currently on the market, the landscape in terms of the drug-resistant mutants, and what you see here is there's problematic issues, especially, for instance, in terms of this K101P mutation, and our compounds. These are just some of the examples, and these are two of the most potent, 506 and 555, and you see that they have not only very nice properties in terms of their potency and their drug resistance profile, but also in terms of solubility. This is ropivrin that I mentioned that's very insoluble, and so they have really very nice properties. And so we really wanted to think about moving forward with this class of compounds. And so this just sort of summarizes some of the advantages thus far. They have very nice resistance profiles, and especially on this K101P, for which all of the current drugs are currently resistant. They have improved solubility, and especially with respect to ropivrin. So we were really excited, and we wanted to think about moving forward. And so we did some further analysis. I mentioned the indolazine structures. Another types scaffolds that we use are the naphthyl scaffolds that's similar to the indolazine, so a 2-naphthyl derivative and 1-naphthyl and a 2-naphthyl derivative. So these are some of the more recent ones, and one of the compounds that's really kind of rose to the top is this 636 compound here, where we have a difluoro functionality. So this compound has very nice properties, and so we really felt like that this really kind of checked all of the boxes in terms of all of the type profile that we wanted to have in terms of pharmacological properties, drug resistance, and so forth, and also in terms of toxicity. The therapeutic window here for this compound is over 200,000-fold between the cytotoxicity and the antiviral potency. And so we really wanted to get more information on this compound, because this was kind of our lead candidate at this point. And so we did some pharmacological profiling with this compound, because we really wanted to look for potential off-target effects. And so this just compares it as well as this other compound, 532. I've listed them here as compounds 1 and compounds 2. And in general, you see that they're very nicely devoid of off-target effects, even compared with the ephedrins, which is currently on the market and used in a lot of combinations. With one of the compounds, there is this inhibition of CYP219, but that's actually an isoform that is not responsible for metabolizing the majority of the drugs. The majority of drugs are with this CYP3A4 that we really worry about. And so we wanted to go further then and get some real pharmacological information in terms of PK. And so this just sort of summarizes the important properties, and we wanted to really go forward and look at how these compounds behave in mice. You know, did they have a long half-life? In order to be a viable drug, it really needed to have a longer half-life. We wanted to at least be able to dose once a day. So we wanted the drug to hang around at sufficient levels so that we could actually get away with doing dosing for one day. So the drug would hang around. And so this is just an example. This is an IP injection in mice. And we were really excited to find that there were significant levels of the drug, very high levels of the drug, at 24 hours and even down to 48 hours. So this is a single dose of the drug. So this started to tell us that we had really nice properties with this, and we weren't going to inject it into the animal and it would just be metabolized really quickly. So we were excited about this, and we compared it with ephedrins and also with our other compound, the 532, which is a cyanovinyl derivative that's also very potent. And it really kind of checks the boxes in terms of its nice pharmacokinetic parameters. So this really told us that it was actually a good starting point for thinking about further development of the drug. And we also looked at the off-target effects, and those also seemed very good as well. And so we went ahead and we did the three-dimensional crystal structure. We hadn't done the structure at that point, and indeed, it interacts with the same types of residues. This is just a three-dimensional structure, the electron density, and these are some of the key interactions as I've illustrated here. And so at this point, we wanted to think further about the selection criteria. What I've listed here are some of the important boxes that we really want to check in terms of thinking about going forward, because we've made a lot of compounds at this point. And so we've checked some of the boxes. One, we know that they have good PK properties. The other thing that we wanted to do is we wanted to ask the question, how does this drug behave with other HIV drugs? Because at the end of the day, it's going to actually be dosed with the current drugs that are on the market. So, and there are examples where there are synergistic interactions between drugs, and those are the ones that would be favored for combination therapy. So we wanted to look at that. We wanted to evaluate them and evaluate the compound in a humanized mice model and think about whether or not we could develop a long-acting formulation of the drug. And so I'll just show you some of the synergy studies that we did, and we were really excited with this compound to find out, it's illustrated here as compound 1, that it has very potent synergy. In these synergy plots, if one sees peaks in the positive coordinate, that actually illustrates that there is synergistic inhibition with each of the different classes of drugs that are currently on the market. So not only the nucleoside drugs, but also integrase inhibitors. So we were really excited to find out that it was not only very potent, but also synergistic. And so at this point, the conclusions were, again, we're checking these important boxes with these compounds, and this potent synergy really told us that maybe we really are ready to think about going forward. So these are just some of the summaries, and we have the right PK, so now we're ready to go into humanized mice and ask the question of whether or not these might have efficacy. We know that if we give the dose, we can actually have the drug hang around long enough to hopefully be efficacious. And so we collaborated with Priti Kumar's group at Yale, who works on HIV, and also she has humanized mouse models to allow one to check efficacy. At the same time, we also wanted to explore the possibility of developing a long-acting formulation for the drugs. So we also collaborated with Mark Saltzman's labs, who's in bioengineering. He's an expert in nanoparticle delivery, so we wanted to also ask if we could make a nanoparticle formulation of the drug and hopefully develop a long-acting formulation. And so what I show you is just the schematic for how the mice were first infected with a clinical strain of HIV, and then we actually analyzed serum samples. We took, these are now humanized, so they're actually reconstituted with the human immune system, the human T cells, and so we looked at the CD4 count as well as the viral load. And what we looked at here was dosing every day with free drug, but we also gave a single nanoparticle dose. So we were able to load the drug into a nanoparticle formulation. And this just shows some of the effects. We can see that the drug actually hangs around with the nanoparticle formulation illustrated here we can actually see in blue that one single dose of the nanoparticle formulation hangs around for almost a month. So we were really quite excited about that, because that would actually tell us that we could give one injection of the drug and it would actually perhaps even be protective and be efficacious. And so we wanted to look then and see if we could test these compounds in humanized mice. And so we do this in two ways. We look for the ability to protect T cells, okay, in the absence, so after T cells are infected with HIV, they're completely defeated, okay, as illustrated here by the black points. And so if we have protection, then we maintain our CD4 levels, and so you see that even out to close to 30 days, we actually protect the T cells, and then also we get a drop in viral load. And so we see that here, so this is full-blown virus, and then in the presence of the drug, either the nanoparticle formulation or giving the drug daily, we see a drop below detectable limits. And so this really tells us, and we're very excited about this, that our drug not only has good properties, but it's also efficacious, not only as a single, as a daily dose, but also as a single nanoparticle formulation. So really, I'm summarizing here then our top 10 list for these things that kind of check the boxes in terms of all of the right properties. There's a lot of stuff on here, so don't worry about it. These just I say that I think we've gotten to the top 10 list of thinking about this compound as a preclinical candidate. And the 11th thing that I just added here, and it's actually added in the last couple of weeks, is we've started to do some HIV implant studies. I mentioned the nanoparticle dose, but what if we could put the drug in an implant, such as an implant that one might use like a contraceptive implant. And the Saltzman lab has actually had a lot of experience with that, so we decided to do some pilot studies. And so we're very excited. We have some preliminary data right now that shows that the drug hangs around for 3 months, so we still see plenty of drug for 3 months, and so we've just started some efficacy studies. So we're excited to see where these may take us, but that would actually be a significant improvement in therapy if a patient only had to have an implant every 6 months or even every year. A lot of the contraceptive implants will last even a year. So that's one of the places that we're going with this, and we're also exploring even more efficacious drugs. Now, just in the last couple of seconds, I want to tell you about a newer area that we've also started, just in terms of thinking about developing even yet newer scaffolds. And one of those scaffolds is actually a scaffold to target drug-resistant mutants, and we actually were interested in developing some covalent inhibitors, particularly against drug-resistant mutant forms of HIV. And so I wanted to just mention some of those studies that are sort of a first step toward developing these inhibitors. And so the idea here is that maybe we could take one of our current scaffolds that we have and use our structural information, and perhaps, especially for this particular mutant, which is a cysteine mutant, this Y191C is a key drug-resistant mutant, maybe we could design this chemical warhead, as I've illustrated here, some of the possibilities, and actually alkylate that cysteine residue so that we would get a covalent type inhibitor. So we got some clues from our functionality for exactly how we wanted to design this compound from some of our compounds that we made in the 2-naphthyl series, as I've illustrated here. We actually looked at this compound, and as I mentioned, with the 2-naphthyl series especially, they're very, this is just an example of this Y181C mutation, they're very resistant. And so could we take something like this, which is good on wild type, but then we have trouble with the drug-resistant mutants once it's much less potent, could we convert that into something that would now be an inhibitor by making it a covalent inhibitor? And so we were able to solve the crystal structure of that particular mutant, and what clued us in, especially with the Y181C, is that this chlorine group looked like it was pretty close to this cysteine Y181, and so we reasoned that we might place that chemical warhead in this position, and that was some of the early pilot structures that we wanted to make as a test of proof of concept for this strategy. And so we made several different types of chemical warheads, as I've illustrated here. So a couple of alpha halo derivatives that would alkylate by this SN2 type attack, displacing the halide, so that's illustrated here. These two type derivatives, and then alpha beta unsaturated carbonyl derivatives illustrated here in 4 and 5, where we would have a Michael addition of that cysteine residue and the alkylation of the enzyme this way. So this was sort of the chemical warhead strategy that we wanted to use. And so one of the first things that one does in looking for covalent inhibitors is one asks the question of whether or not they're time dependent. And so that was one of the first tests that when we made these compounds that we wanted to ask, do we see time dependent inhibition, and is that time dependent inhibition in the presence of the drug resistant mutant? And so this just goes to show you what happens if we incubate each of these compounds for a longer period of time with the enzyme, and you see that two of those we actually start to see a decrease in enzymatic activity, and that's actually because they're actually alkylating the enzyme. So you see a shift in the IC50 value. It gets more and more potent as a function of time, and that is actually primarily in the context of the drug-resistant mutants, the Y181C. So we see that we have this kind of behavior where we improve the potency with the Y181C single and double mutations. So we see that we improve it to down into the 100 nanomolar range. So this told us that we were really in the right place, but we really wanted to validate that we do indeed have alkylated these positions. And so we used mass spectrometry to try to show that these compounds are indeed acting only on the P66 subunit in that allosteric pocket. And so what we did is we sprayed the full-length protein, and we looked at the P66 and the P51 subunit with each of these compounds. And what we see in the case with 3 and 5 is we see a shift in mass only in the presence of compound 3 and compound 5. So we see this mass shift, and that tells us that there's only one molecule of this, of each of these compounds bound to the P66 subunit. So that very strongly implies that it is indeed covalently modifying it. But it's this type of situation where this is a lot of experimental evidence, but really a picture is worth 1,000 words. And so we really wanted to determine the three-dimensional crystal structure, and so we were actually able to do that in each of these cases. And what one sees here is very nice electron density. That was the predicted cysteine that was going to react, and so we can see a nice conductivity in electron density, and these compounds are indeed bound in that allosteric pocket as we had hypothesized. And then finally, we looked at these in cell culture, and we showed that they're also potent in cell culture and even have a slightly lower IC cytotoxicity than some of the current compounds like ephedrins and rilpivirine. So all of these really suggest that this is another important class of inhibitors, and so we wanted to think we're actually further improving these, but this is a very new strategy that we've started, and I think it could also yield some very interesting and exciting new compounds to think about for treatment. So I'd just like to finish up now and kind of summarize what I've told you and some future directions. I wanted to give you an example where we've used a computationally guided and structurally informed strategy to make potent non-nucleoside drugs that have improved pharmacological properties, and they're comparable to the current FDA-approved drugs. We've shown that they have efficacy in an animal model, and we've actually had a long-acting formulation that has efficacy in an animal model. And we have implant studies that I mentioned that we're quite excited about. And we have the first covalent inhibitor of reverse transcriptase that destroys the drug-resistant form. And so now what we're after is developing even newer compounds that also act on the wild-type HIV. So I will summarize that, and then most importantly, thank all of my collaborators, the folks in the lab who actually did the work, the folks in my RT as well, RT group, as well as former members of the group. My collaborators, Bill Jorgensen and his group at Yale for the computational and synthetic studies, Priti Kumar's lab for the humanized mice work, and Mark Saltzman's lab for the bioengineering stuff, and then we also got some of the drug-resistant screening from Bob Silicano's lab at Johns Hopkins. And I'll stop there, and thank you for your attention, and answer any questions that you might have.
So, Karen, I wonder if you could share any thoughts you might be aware of that makes HIV uncurable. Because hepatitis C can be cured by treatment. That's a virus, right? And so what is it, if it's known, about HIV that requires lifelong treatment?
Yeah, so remember, so it's a retrovirus. So the DNA gets integrated into the host cell genome. Okay, so all of your cells now have the integrated viral DNA. And so the problem is that we have a whole population of latent T cells, and there's a lot of debate in the literature as to where those cells are hanging out. But basically, they have this viral information, these cells, and they're actually in a latent state. And so how do we treat that? Do you try to reactivate the virus? There was this theory called the shock and kill. So basically, you try to get an agent that would reactivate the virus. This is not the situation for Hep C, for instance. And so the question is, how do we target those reservoirs that we think are in the lymphatic system? And it's a very slow-growing, resting population of T cells, memory T cells, that we need to get rid of. And that's really the challenge, is how to get, are we just not getting drugs in? There's certainly strategies to use higher concentrations of drug, but at the end of the day, we still don't get, if you take a patient off therapy, the HIV will come back. You might have heard about a case a couple of years ago called the Mississippi Baby, where they treated the mother with a very high dose. So the whole idea was to completely suppress the virus during birth. And so the child, like for a couple of years, showed no signs of HIV. And so people were really excited. They thought this might be another cure. But then once she got to like 4 or 5, again, think about this population, this very slow growing population that eventually virus started shedding from the lymphatic system. So that's really the challenge, is to try to understand how to get at this population, how to get drugs into wherever that, anatomically, wherever that population may be, and rid those cells, And there have been this shock and kill strategy where you actually give a drug that would reactivate the virus, and so the whole idea is maybe you can clear it. Those strategies in clinical trials have not really worked thus far, so there's still a debate in the literature as to how we can actually develop a cure. But hopefully we'll get there. But I think we have a lack of understanding right now for how to actually do that. Yes?
What's known about the rate of the formation of the HIV's ability to make those mutants?
Oh, how quickly that occurs?
Quickly is it-
Oh, when nevirapine was the first in an RTI, I'll give you the results in a patient, okay? There was a lot of excitement about these drugs. They went into the clinic, and again, this is before we got smart enough to think about combination therapy. So they went, the non-nucleoside nevirapine went in the clinic, and they looked very potent, okay? And so they were monitoring virus. Within a month, they actually developed drug resistance. So that one Y181C mutation that I showed you, that developed within a month. So that's the problem with single agent therapy. You actually have to use multiple strategies for therapy and try to keep the drug resistant population low enough with the multiple agents that you don't get what we call viral breakthrough.
Is there much known about mutations of the proteases, or is it mainly just?
Certainly the same kinds of things happen with the proteases as well. And in fact, there are different cocktails of therapy that would be a mixture of a reverse transcriptase inhibitor combined with a protease inhibitor. But the same kind of thing, especially, this is sort of the bad thing about targeting an allosteric site, is that it's much easier to develop resistance. Like if you talk about a nucleoside analog, it takes more like 6 months to a year to develop resistance. And that's because you're targeting the active site. And so that was actually one point that we were quite interested. I should have pointed this out. Our compounds actually interact with, in that pocket with proline 95 and tryptophan 229, and we know that those are immutable residues, so we don't find those as drug-resistant mutants. And so if you can potentially target those, and those are key interactions, and if the virus, if it's detrimental to the virus, then maybe it will be, it won't mutate. But just in experience, there will be resistance, and so one has to try to stay ahead of the game. Yeah.
How do you feel about how well you can stay ahead of the game with the time it takes to develop a drug and the ability of these things?
Well, I think that the hope is that, and actually when you go to the, if you go to the FDA for even asking for drug approval, you really need to know what, and this is actually something that we're doing right now in the laboratory, is we're putting our drug in to try to figure out what our drug-resistant mutants are going to be, where our compound to, make it further clinically. We need to kind of know that. We need to have our plan for, what would be next. And as I mentioned, it's well described as trying to hit a moving target, try to stay ahead of the game. So. Yeah.
Can you elaborate on nanoparticle formulation, like what that means exactly? I don't really.
I'm sorry, say
Nanoparticle formulation? I don't really know what that is.
So with the nanoparticle formulation, these are PLGA nanoparticles, so they ultimately, that nanoparticle will get degraded. So the nice thing is that PLGA nanoparticle is already approved by the FDA. And so one gives it as an injection and over a very slow period of time. So the drug gets released over a slow period of time, and then the nanoparticle itself is gradually degraded, and it's degraded into glycolic acid, so natural components. So there's a lot of history for that kind of material, that it's biocompatible and metabolized over a longer period of time. So that's one of the reasons that we started with the PLGA-based polymeric type. So it's known as a nice drug delivery type system that's been used before. Yeah.
So what's known in terms of trying to control this virus globally in sort of third world settings, developing countries? Because if we're generating resistance so fast, if we are using a single drug or perhaps the wrong drug regimen, like who controls what drugs are given where, and how is that monitored, and who oversees that?
The good news is that we never give a single agent. Actually, only primarily for PrEP, for pre-exposure. That's the only time that we would use a single agent. So all of the combinations that are approved now are either at least 2-drug combination, often a 3-drug combination. And even in developing countries, they're the cheaper drugs, like for instance the nucleoside drugs, and like one of my colleagues, he's actually passed away several years ago, but D4T was one of the nucleoside drugs, and they actually at Bristol Myers Squibb developed that drug. They decided to release the patent restrictions in third world countries to allow that drug to be one of the treatment regimens. So I think the statistics are now that in developing countries, about half of the people are treated. So that's actually a step in the right direction. We would like for that in the next, decade or so to be, higher than that. But it's a big step, I think, and that kind of, there's a lot of surveillance going on. I think that the kind of, to me, scary and disturbing part about HIV is that even in the United States, there are 1 million people that don't know they're infected, and especially, there's been a resurgence of IV drug use. You might have heard a couple of years ago about this whole county in Indiana where there was this big outbreak, and again, with among IV drug users. So those are places where we would really, at the very least, like to have PrEP, pre-protection for HIV. And so I think there have been a lot of strides, but the goal would really be. Even now, we will still have over 36 million people that need to be on therapy for the rest of their lives. So that really covers. Unfortunately, we have a lot in our research to do to even cover those people, until we find a cure. And so that's sort of the problem. Certainly, the epidemic has slowed down, but we need to really think about therapy for these patients. In order to have the quality of life, they need to take the drug. They still need to be on drugs until we find a different strategy. So what we've been trying to do is improve the regimens. Like if we develop an implant, if you only had to have an implant every 6 months or a year, that would be much better than having to get up every day and take your medicine every day, even if you don't feel like it. You have to take it every day, and that patient compliance is, if you talk to the clinicians, that can be a factor. So those are types of things in the field that are ongoing to try to improve those sorts of things. Other questions? Yeah.
With your when you're doing your transient kinetics experiments, in the one that I think is when you're doing stop flow there, I've heard of where they're measuring the rates and stuff using absorbance, but you were saying that you were
and those are rapid chemical points. So we're actually using radiolabeled. We're looking at radiolabeled conversion of radiolabeled DNA to the elongated DNA. So in experiments that I showed today.
So yeah, I was gonna ask, like, what's the purpose in chelating it with EDTA at the end?
Because the site, in order for polymerase catalysis to occur, you need the metal ion there to properly orient the aspartic acid residues. And so if you pull out the metal, the catalysis won't happen. So that prevents catalysis. It just very quickly pulls out the metal and catalysis stops. So that's sort of the strategy.
Thank you.
Yeah.
How flexible is this hydrophobic binding pocket? Because you see a lot of these RT structures with these NNRTIs, and they kind of they space fill around the drug.
Yeah. So RT is notoriously known to be flexible. In fact, you have to cross-link the DNA to get a structure. And so there's one paper that actually described it as like a gymnast, you know, doing handstands. And it's historically known to have a lot of flexibility. And so it makes the crystallographer's job a little bit challenging, as compared with other polymerases where you can perfectly crystallize them without cross-linking the DNA. That hydrophobic pocket, interestingly, if you just saw the apo structure, that hydrophobic pocket does not exist. So that's what's really interesting. It's only with that first structure by Tom Seitz where he showed that allosteric pocket. No one had any idea that that pocket would even be there or why it inhibited. So that was kind of how we got into the whole area with Tom's structure, is to try to figure out, well, how is that? That's way over there. How is that actually inhibiting catalysis? And it's 10 octaves away. So that's kind of what sucked us into the problem initially, is just the mechanistic, trying to understand how that works. And so it's a nice example where it was actually that compound was discovered with a high throughput screen. And then when they looked at it, they're like, you know, this doesn't look like a nucleoside analog, you know, how does this work? And so that's sort of how that pocket got discovered. Yeah?
What the drugs that you've created, how does that compete with like the cost of what's already on the market? Like, is it more expensive than the ones that are like synthesized?
Of course, yeah. I mean, I guess I would say that with the HIV, like the hepatitis C drugs, for instance, I teach a lecture to the medical students, and it's just kind of scary. A year course of therapy is about $80,000 for Hep C. So in that landscape, they're cheap. But the problem is that with Hep C, you know, it's can be curative. Whereas with HIV, you know, you still have to dose it every day, you know, and take it chronically. So obviously, the newer drugs are always more expensive because, you know, with the model for pharmaceutical industry, they need to recoup their costs. But there are other models now for, you know, trying to repurpose compounds and trying to make things cheaper. And, you know, I think maybe going forward, especially with, this is also probably more relevant for antimicrobial agents, we don't really have enough stuff, drug discovery going on in those areas. All of the big pharmaceutical companies have moved out of it because it's not as profitable as these expensive things like immunotherapy and hepatitis C, for instance. So, you know, it's a tough landscape in that regard.
Well, with that, I'd like to thank Dr. Anderson for joining us. Feel free to come to talk tomorrow and the other events that are planned. So, thanks.